We'll just wait for another minute.
Yes. Good evening, everyone, and welcome to this webinar of IPEC India in association with Azilis and DSM for the benefit of the attendees. Let's keep the headphones and earphones connected for better clarity. Questions, if any, can be incorporated into the Q&A section, which will be attended to. In case, due to the time limit, we are unable to attend any questions, you can email them to admin@ipecindia.org and we will respond to them promptly. Let me introduce Dr. Rajesh Parab. He is an honorary treasurer of IPEC India. He has completed his M.Pharm in Quality Assurance from Goa College of Pharmacy and doctorate in Pharmacy from Goa University. He has close to 30 years of experience in various functions of pharmaceutical industry that include general management, plant operations, technical services, regulatory services, and formulation development, to name. His career spans working experience at companies like Ciba, Knoll Limited, which is Abbott now, Astra, Banner Pharma Caps, Forum Bioscience, and latest at Calacon Asia. His latest position was Managing Director, South Asia and Global Leadership Team member for Calacon. He is recipient of Lean Six Sigma Green Belt, ET General Manager Award in 2019. Dr. Parab is also contributing to larger communities through his involvement in managing committee of various organizations. He is a board member for IPEC India. He is a member of for higher education in pharmacy at Goa University, member of management for Goa Pharmacy College, member of governing body for Goa State CSR Authority. His strong leadership qualities and people-centric approach makes him a successful and popular leader. Dr. Parab has effective problem-solving and result-oriented style of functioning that make him an indispensable leader and invaluable asset to any organization. He likes reading, traveling, and listening to music. Over to you, Dr. Parab.
Thank you, Prema. Hope that was not too long. Okay. I'm audible to everyone, hopefully. So, distinguished members, participants, speakers, ladies and gentlemen, good afternoon, good evening, and good morning. On behalf of the International Pharmaceutical Excipient Council of India, I am pleased to welcome our guest speakers of today, Dr. Ann, Dr. Fernanda, Dr. Misek, and all participants joining from different parts of the world, as well as colleagues from India who are representing the industry as well as academia. We welcome you to this webinar organized by IPEC India jointly with support from Azilis and dsm-firmenich on the topic ascorbic acid and α-Tocopherol as nitrile scavengers, an elegant and simple solution to mitigate nitrosamine in drug products. I believe this is one of the current hot topic where everyone is most like to learn and share what knowledge they have. I could see that many of the participants are not members of IPEC India and hence may not be familiar with the IPEC activities. Hence, I would like to take a few minutes to give you a brief about the IPEC activities. The IPEC Federation is a global association that serves the interests of producers, distributors, and users of pharmaceutical excipients. IPEC currently is a federation of 5 independent regional industry associations. It's headquartered in the United States. Then there's Europe, Japan, China, and India, with total global membership extending to more than 200 companies. It provides a unified voice to promote the best use of excipients in medicines as a means of improving patients' treatment and safety. IPEC India is a non-profit organization with the objective to develop and promote voluntary guidance and other programs for the world pharmaceutical industry. These are designated to ensure continued availability of excipients and related components to finished products that meet the highest appropriate standards. for quality, safety, and functionality throughout the manufacturing process and supply chain. Importantly, to educate and cooperate with regulatory authorities, industry organizations, and scientific bodies working to advance public health on matters relating to the manufacture, distribution, use, and functionality of excipients. To develop a promotion and communication program by means of seminars, webinars, workshops, participation in symposiums, exhibitions to inform government, industry, media, and the public about excipient issues and accomplishments. To create awareness among excipient manufacturers, distributors, and users about the various IPEC guidelines which help the industry to achieve the same goal. To create standards and procedures towards self-regulation to assure quality, safety, and integrity of natural pharmaceutical foods, products, services, and labeling information. The key objective of IPEC India is to bring all the stakeholders on a common platform to understand each other's requirements and work together. I'm confident that the excipient industry in India, with its continued and consistent innovation and research, can lead to enormous benefits. I'm sure that the presentation by distinguished speakers from dsm-firmenich will be highly enlightening and thought-provoking. Thank you and have a knowledge-sharing evening.
Thank you very much, Dr. Parab. I'll get the session started, and it's really a pleasure to be here. Thank you for giving us the opportunity to talk about this very hot topic, very important topic of nitrosamines. I have the pleasure to moderate this session that is going to be also delivered by Anne-Cécile and Zdravka. They are experts in this topic. So I'll introduce Anne-Cécile and Zdravka, and then I'll share the slides. So I'll conduct the slides for them. Anne-Cécile is our global science and innovation lead at dsm-firmenich. She's got a PhD in molecular pharmacology and toxicology from the University of Southern California. And she leads our innovation projects involving our vitamins, nutritional lipids, and flavors for the pharma and medical nutritional segment. And Zdravka is our principal scientist in innovation. She's got a PhD in pharmaceutical sciences from the University of Basel in Switzerland, and her research is focused on the evaluation of the performance of vitamins and other nutritional actives in oral solid dosage forms. And my name is Fernanda. I'm the Global Market Development Manager for our pharma excipients portfolio at dsm-firmenich. So let me share my screen, Dr. Parab, and we will get started. I trust that you can see my slides.
Yes.
Yes, we can.
Okay. For some reason, it's showing as black here for me, but I guess it will come in a minute. Before we get into the slides, I just want to say a couple of words about dsm-firmenich. So actually, the company was formed in May 2023 after the merger of two big companies, DSM and Firmenich. We are a global company, but we have Swiss-Dutch headquarters. We reinvent, manufacture, combine vital nutrients, flavors, and fragrances for the world's growing population. We are part of the Pharma Solutions division, and our portfolio includes vitamins, carotenoid and cannabinoid APIs, lipid intermediates, and specialty excipients. So with that, I will hand it over to Anne-Cécile and Zdravka.
Thank you, Fernanda, for the introduction. I hope you're all hearing me well. Hello, everyone, and thank you for attending this webinar. The objective of today's session is to share some information about nitrite scavengers of use of nitrosamine, and two, to share key aspects of formulation with ascorbic acid and α-Tocopherol. But first, let's start with a short introduction to the topic of nitrosamines. If you can go to the next slide. Fernanda, can you go to the next slide?
And there is a little voice disturbance.
Yep. You can hear voice
And
disturbance.
Cecilia, your sound is breaking up.
Yep.
And Cecilia, your sound is
My
breaking up a little bit.
sound is breaking up. Can you hear me better if I put the microphone closer to me?
That was a little better.
Okay. Hopefully this will not break up too much. So yeah, so the need to evaluate the risk of nitrosamine formation, measure it, and mitigate it is becoming an integral part of all product development. And while the limits of nitrosamine levels will keep on evolving as the evidence for safety and carcinogenicity of nitrosamines accumulate, keeping nitrosamine formation under control is imperative. And the FDA in the United States has been leading the way in setting a framework to deal with these impurities. Now, nitrosamines are a chemical reaction between
Dr. Anne, it's a little jerky.
amine. Yep.
Yes, the sound is not coming smooth. It's jerky.
Yep.
Try removing your headset or using the computer mic.
I can remove the headset. Let's try this.
Try that.
Can you hear me better now?
It was better. Let's see.
Is it better?
Yes.
So I will do without the headset. So as I was saying, N-nitrosamines are formed by a chemical reaction between secondary or tertiary amine and a nitrosating agent. And while these compounds have been known and studied for decades, and in fact, the link with cancer dates back to the 50s. Now, the condition for nitrosamine formation can be met at many steps during the production of drug product. What you see on the scheme here from the left side, from the drug substance themselves to the excipients, the solvents, the water, the manufacturing process as well, the packaging and the storage. It can all create conditions for the generation of nitrosamines. And many strategies can be used to deal with nitrosamine formation. For example, and I'm sure by now you're all using this kind of tools with like a strong qualification program for all the compounds you use in the manufacturing to try to keep the level of nitrosamine precursors as low as possible. And there are other ways. But today we are focusing on one of the strategies drug developers can use to keep nitrosamine in check in their drugs. And it's based on the use of nitrite scavengers to reduce or stop nitrosamine formation in your product. Next slide, you'll see that there are many nitrite scavengers able to do the job. And Fernanda, if you can go to the next slide, we'll see them. So some of them are listed on the slide you will see in a second, but they are not all equal, however. Some of them are more active than others or more appropriate for one form over another. Some of them don't appear on the inactive ingredient database of the FDA and as such are not so attractive. Others do, but don't appear to work in solids. But two of them, which have consistently given good results for nitrite scavenging abilities, are ascorbic acid, especially, and α-Tocopherol. And on the next slide, what you see is just a way to illustrate how these two molecules block nitrosamine formation. Nitrite, which you see on the left here, are impurities in nitrous acid, HNO2, or nitronium ion, H2NO2 plus, under acidic conditions. They'll further transform into other nitrosating agents, which will react with secondary or tertiary amines and form nitrosamines that you don't want to see in your product. Now, both ascorbic acid and α-Tocopherol interact rapidly with nitrite ion and as such stop the process of nitrosamine formation by stopping nitrosating agent from being formed, basically. Now, a few words about α-Tocopherol and ascorbic acid. So ascorbic acid, which is also known as vitamin C, is best for moderate to strongly polar environments, while α-Tocopherol, or vitamin E, has more affinity for less polar and non-polar environments. Ascorbic acid is therefore more suited for various delivery systems that range from solutions, injections, tablets and capsules, and α-Tocopherol mostly used for creams, ointments, and tablets. Now, what you see on the following slide is a result obtained by Dr. Nenda and his team in 2021, where they looked at the inhibition efficiency of ascorbic acid, sodium ascorbate, α-Tocopherol, as well as ferulic acid and caffeic acid in a solid model drug. And they saw that these molecules were effective in these constrained conditions. Now, this was very encouraging, as it showed that even in the condition of a tablet manufacturing, the addition of nitrite scavengers could be beneficial. So since then, a number of publication and presentation at various meetings have reinforced the idea that these scavengers are safe and useful to keep nitrosamine formation in check and keep drug product as safe as possible. And that's what you see on this slide. On the efficacy side, the work of Omshak and colleagues, as well as the work by FDA and GSK, has added data showing that ascorbic acid in particular works well in tablet form. On the potential impact of drug absorption, Dr. Chris Bode from Pharmaron Lab Services has shown no impact on bioequivalence for and on the toxicity and safety topic, of course, both ascorbic acid and α-Tocopherol have a very long history of safety use in products. Internally, as extended this experimentation.
Your sound is not clear, Doctor. I believe there's some network challenge. You are not sounding very clear. I don't know whether I'm getting that, others also.
All right, so I mean, hopefully
now it has changed, but sometimes it stops.
Yes, it's breaking. Your voice is breaking. Yeah.
Right. But we're going to move to Zratka. So in a minute now, she will share, and hopefully her voice will not be broken up. But we'll share the important piece around ascorbic acid and α-Tocopherol in the experimentations we've done ourselves. And now Zratka.
Thank you, Anne-Cécile, and I hope my voice is better. Please let me know if this is not the case.
You're good.
So, is it working?
Yes.
Thank you. So I will say now a few words about the case studies where we have evaluated the impact of ascorbic acid on nitrosamine formation. We have tested it in solid dosage form, and then we have also checked how α-Tocopherol can affect the nitrosamine formation on in gels. So on the next slide, I would first like to say a few words about important properties of ascorbic acid when it comes to formulation. Not all ascorbic acid powders that are on the market are the same. The very important property is particle size, and the products on the market can differ in particle size. On the left graph here on the screen, you see particle size distribution of various products on the market. They are from the ultrafine powder up to the much more granular standard grade, up to 600 micrometer in particle size. Why am I emphasizing the particle size? It is because this can impact some properties of the molecule itself. For example, ultrafine powders having a small particle size will have a large surface area, which can be reactive, so it can have much higher antioxidant property comparing to the larger particles with then overall small surface area. As well, also important property of the ascorbic acid are impurities. And on the right-hand side of the slide, you can see how these impurity levels can differ in different products on the market. On the next slide, if we can go further, Fernanda. Thank you. We will see another property of ascorbic acid powder, which is relevant for the formulation, and that is morphology. So scanning electron microscopy pictures here can show you different morphologies on the products of the market. They range from the cubicles to plate shapes, irregulars, and this is quite important when it comes to processing, for example, flowability and compressibility into the tablets. It needs to be taken into consideration. On the next slide, we go to our case studies. So here is a study where we have tested how nitrite reduction, namely we have produced placebo tablets without active substance. We have used direct compression and wet granulation as manufacturing routes. Those tablets were containing different levels of ascorbic acid in them, so 0.25%, 0.5%, and 1%. And the tablets after production were stored in accelerated stability, so in chambers for 7 days at 40 degrees and 75% relative humidity. What is important to note here that the ascorbic acid here that we have used, this is ultrafine powder, it reduced nitrite content in the tablets, and either if they were produced with direct compression or wet granulation. And remember from what Anne-Cécile was saying, if you are reducing the nitrite levels, then you're also blocking actually the nitrosamine formation later on. Generally as well, what we can say is that the higher the ascorbic acid concentration in the tablet, the lower the nitrite content was measured. On the next slide, we have another case study. And in this case, we have checked the formation of nitrosamines, in specific N-nitroso impurity of a certain model drug was tested. We have produced tablets as a control without ascorbic acid, and also tablets with ascorbic acid at 1% level. We have spiked the tablets with potassium nitrate, and the tablets were produced with direct compression. After the production, the tablets were stored for 2 weeks at 50 degrees and 75% relative humidity. And we can see on the graph that the tablets as a control without ascorbic acid contained, after only 2 weeks, high levels of N-nitroso impurity, like almost five times higher than at the beginning. And the tablets that had 1% of ascorbic acid in them have barely seen the increase in those N-nitroso impurities. So this is a real case, showcase where the nitrosamine formation is prevented by using ascorbic acid. On the next slide, we will also present a case study with another 2 model drugs. In this case, the tablets were produced using fluid bed granulation. And those model drugs were different in structures, contained both secondary and tertiary amines. And they also had the different FDA limits on nitrosamine content. Both of those tablets contained 0.5% ascorbic acid. And the way we produce them is schemed here. So we have added ascorbic acid at level of 0.5%, as I mentioned, dissolved it in water, and then use this solution as fluid bed granulation solution together with other excipients. Stepwise API was added, and then at the end the lubricant as well was added to this tablet mixture. And after that we tableted the mixture. So on the next slide, we will see the results of this study. So the tablets were put on 40 degrees and 75% relative humidity. We have observed the API-related nitrosamine levels over 3 months. You see for both model drugs. And if we go to the left side of the slide, we'll see how the increase of it's a blue line, of the API-related nitrosamine was observed. It was quite steep increase, and the dotted red line is showing what is the FDA limit for this drug compound, and what they set up as acceptable. So the graph shows us actually if you if you add ascorbic acid, this is the orange line, we have clearly seen no increase in this impurity over 90 days storage. So this clearly says that actually if you are using ascorbic acid in this formulation, you will be safe when it comes to this nitrosamine impurity. The graph on the right side of the slide is showing another model compound, and the tablets were also stored at 40 degrees and 75% relative humidity. Here we see the same with blue line increase of the API-related nitrosamines, and increasing quite fast. I mean, after 2 weeks, we see very, very high increase. And when we have added ascorbic acid at 0.1%, we saw no increase of this API-related impurity. The red line shows about what is the FDA limit for this drug compound, and it is clearly seen that already shortly after production, without ascorbic acid, these levels are higher. So we can conclude that actually adding ascorbic acid for these drug molecules and this formulation was very successful and effective in reducing and blocking nitrosamine formations. Let's go to the next slide, where we talk a bit about discoloration. I'm sure anyone working with ascorbic acid has observed this effect. However, this is something that is quite known when it comes to using ascorbic acid, and can be mitigated in some cases. For example, also you see the pictures here of tablets that we have used in these case studies. The picture of the tablets on the left those tablets contain ultrafine powder ascorbic acid, and this ultrafine powder is showing slight discoloration because the particles are so fine. So actually the shade of the tablets is minimally changed. And when you are using the larger particle size ascorbic acid, then some spots of the discoloration can appear. These tablets contained 1% of ascorbic acid, and they were also exposed to the harsh conditions for a month at 40 degrees and 75% relative humidity. So on the next slide, we see also pictures from the studies I have previously mentioned. It is as well, so there we have followed the impact on the nitrosamine formation. And here we see some minor discoloration of tablets that occurred over harsh storage conditions. So we see tablets with after 1 month at 40 degrees and 75% relative humidity, including this 0.5% ascorbic acid. There is some minor discoloration when it comes to changing color into the slight yellowish, and also within the tablet there is some shading. On the next slide, we see also tablets that were used for the model drug 3, and here is as mentioned as well some shading and minor discoloration observed on the surface as well in the inside of the tablets. So on the next slide, I think we can move to the other part which we wanted to mention, and it's about α-Tocopherol, another antioxidant that can reduce the nitrosamine formation, and in this case is topical gel, so another delivery system. We see on the next slide first the properties of α-Tocopherol. It is highly lipophilic compound. It's clear, colorless, and highly viscous oily liquid. It is practically insoluble in water, freely soluble in acetone, ethanol, ether, and vegetable oils. I would like to point out here that actually, using it for and producing gels, these semi-solid delivery systems will not be that challenging because it is a liquid. It can be easily added to the oil phase of for example, of the emulsions. However, if you want to produce a solid delivery system like tablets, the α-Tocopherol would need to be dissolved either in an organic solvent or water, a mixture of water and organic solvent, and then, for example, granulated together with other excipients. α-Tocopherol is also sensitive to air and light, so it needs to be in airtight containers and protected from light when it is stored. So on another slide, next one, we can see actually how we set up the trial when we have included α-Tocopherol in our model gel form. We have tested as API diclofenac, quite a known anti-inflammatory drug, and produced diclofenac gel containing 2% of diclofenac. We were following N-nitrosodiclofenac and nitrosodiethylamine in the gels when after production. We also wanted to see other physical properties of the gel, like color change, pH, and viscosity. The levels and concentrations of α-Tocopherol we used were 0.05% and 0.2%. And here is also the description of the process, how we produced the gel. On the next slide, we will see the results of this study. On the upper two graphs, there is increase, and there are results for the N-nitrosodiclofenac as impurity in those gels. The gels were stored at 25 degrees and 60% relative humidity, and then also 40 degrees and 75% relative humidity for 4 weeks. And on the upper two graphs, as mentioned, we see the blue lines. Blue line is for reference without α-Tocopherol. There is increase in impurity at room temperature, very clear. And then when added tocopherol, 0.05% and 0.2%, those levels remain very, very low. You see it's ppms, it's about 1 ppm. On the accelerated conditions at 40 degrees, 75%, we have seen the increase of the N-nitrosodiclofenac as well at the beginning, and then decrease. So what does it mean? The N-nitrosodiclofenac as a molecule is quite unstable. It degrades. However, unfortunately, it doesn't degrade back to the diclofenac, and that you are off the hook when it comes to impurities. It degrades into other nitrosamine molecules, and in this case, it can be really seen, for example, on the 2 slides below, on the 2 graphs below. So NDEA, nitrosodiethylamines, at room temperature, the blue lines are high, so it's increasing, and at 40 degrees, it's increasing even higher. So what actually happened is that the N-nitrosodiclofenac can degrade on different pathways, and one of them is definitely turning into the NDEA. And this is what we have seen also, where our N-nitrosodiclofenac has over these hard storage conditions has transferred into the NDEA. And for the NDEA graphs, we see that when upon adding α-Tocopherol at 0.05% level and 0.2% level, we see the impact of lowering those impurities in the gel. So both, this is an indication and quite strong, we would say, even evidence that the α-Tocopherol is effective as nitrite scavengers in gels. On the next slide, we see some more, results of the other properties I have mentioned we followed in this study. So this is viscosity and pH. They remained unchanged through the course of the study over 1 month, where we have followed the changes of the gel. And the different temperatures, 4 degrees, 25 degrees, and 40 degrees, they remained quite stable, those properties, depending on the inclusion level of the α-Tocopherol. Odor remained also unchanged. Color was stable at 4 degrees and 25 degrees for the full month of the study. And when it comes to 40 degrees at 4 weeks, color should be monitored later on, because these are quite strong conditions even for the gels. So one should pay a bit attention with applying correct and good, for example, packaging to make sure that things are that the product stays stable. So I think with this we close the chapters on the case studies, and we can go to concluding remarks and back to Anne-Cécile.
Thank you very much, Zdratka, for your presentation on the work we've done internally to elaborate on the properties of ascorbic acid and α-Tocopherol. So if you go to the next slide, in summary, nitrite scavengers like ascorbic acid and α-Tocopherol can be useful tools when delineating a strategy to keep nitrosamines below the established limits by the regulatory bodies. Use low nitrate levels, APIs, of course, and excipients as much as possible, and consider ascorbic acid and α-Tocopherol as a safety net when you need one. In our experience, very low level of nitrate scavengers, below 1%, can have a very significant effect. And so go with the lowest particle size of ascorbic acid you can find for direct compression to ensure close contact with nitrate sources. And if you have the capacity, of course, you can also try different particle sizes and see what works best for your specific situation. Of course, all situations are different, so checking the compatibility between your excipients and the rest of the formulation should be done early on. And with that, I will hand over to Fernanda to close.
Thank you, Anne-Cécile. So just the last slide before we go into the questions. We do recommend you formulate a safety strategy to mitigate nitrosamines in your drug product, even if you think you have the excipients and the impurities level under control. Like we like to say, you can never be too safe. And ascorbic acid and α-Tocopherol have known records of being very effective scavengers, so they can be very effective on this process of mitigating the nitrosamine formation. And we also, highly recommend you to check out the IPEC position paper on this topic. It's got a lot of good information, a lot of good guidance from the IPEC organization. Thank you. So with that, I will perhaps conclude our slides. We've got Zdravka and Anne-Cécile here for the questions. I will stop sharing so I can see everybody, and I'm getting the questions here. Please feel free to put your questions on the question and answer chat box, and we'll get them. Whatever we can't get to today, we'll certainly get back to you as a follow-up. Okay, so let me do that. And we have a lot of questions that were already coming in as you were speaking. There was a couple of them on tocopherol, on vitamin E. So Zdravka, I think this one is for you. I think you answered this question, but let's make sure that all the information has gone through. Somebody asked how to use it because it is a viscous liquid. So what's the best way to use it? And can you use it as well in tablets? Two questions in one for you.
Thank you, Fernanda, and thank you for this question. Let's try to clarify it a bit. It is, as mentioned, oily viscous liquid. So adding it to a tablet mixture and solid dosage forms is a challenge. It needs to be pre-processed. So also it cannot be just easily dissolved in water because it's not water soluble. So one should have at least a mixture of water and organic solvent for α-Tocopherol. And then this solution can be used as a granulation solution, for example, together with other excipients you're going to use for your tablet production. And in that case, you are incorporating it in tablets. What we can say is that since it is a little bit difficult to be processed and added into the tablets, it is much rarer used for the products on the market. In contrast, ascorbic acid, for example, which is a solid, can be also either very easily dissolved in water and then processed with other excipients, or can be just simply added to the tablet mixture, is more used. And you will see many products on the market already containing ascorbic acid as antioxidant, this case doesn't have to be for nitrosamines, of course, they are using it to protect the drug substance, but from the processing perspective, it is easier to use ascorbic acid in these cases.
Okay, thank you. There's a few questions on discoloration and if there is a way to avoid discoloration, to prevent discoloration. There's been a few questions around that. Can you elaborate a little bit more on the discoloration topic?
Zveka, you want me to take this one?
Mm-hmm. Yes.
All right, so yeah, so I mean, it's definitely not uncommon to see some discoloration appear when storing tablets containing ascorbic acid and under high stress in particular. So the browning reaction itself between the carbohydrate and the amino acid has been known for a long time. So there is quite a bit of information in papers out there. And ascorbic acid has been both, widely used as a single component medicine and as a component in complex pharmaceutical preparation. And so no safety issue has been identified, but it's something, of course, you should monitor. If your tablets are coated, it's going to be less of an issue. If in the food industry as well, the coloration has been studied extensively, but always with ascorbic acid, it's oxygen, temperature, humidity, and what's going to be around it will have an impact. So it depending on the situation, you'll have more or less. But yes, if you control parameters or are very much aware of those conditions with humidity, temperature, and oxygen, you can try to control it this way. You can coat as well, and so of course the longer time, the more color. But it also, in the case of use as excipient in pharmaceutical drug product, you're talking about a very low level of inclusion, and you should always, of course, try to go for the lower level, and you'll have also far less issue with coloring. So we've always stayed below 1%, and that's where you should be. And we see a great efficacy already at that level in terms of nitrosamine mitigation.
Thank you.
And for whoever asked the question, of course, if there's a need to share some papers, we'll be happy to follow up and send some information regarding the browning.
Another one on ascorbic acid. What would be the impact of particle size of ascorbic acid on nitrite if it is dissolved in solution?
Yeah, maybe I can take this up. So of course, the particle size of ascorbic acid will have an impact only if it is added as a solid form. If you are just adding it directly to other excipients and then API and go for direct compression, then the particle size having, for example, ultrafine powder with a huge surface area will make an impact. And there are already some data on it showing that the antioxidant effect of those small particles with big surface area are much more powerful than bigger particles with less surface area. If you decide to go for solution of ascorbic acid, then you are doing a granulation anyway for a different reason, it would then not make a big difference if you're having a smaller particles or bigger particles, because the solubility of ascorbic acid in water is quite high. So from that perspective, it doesn't play.
Thank you, Zdravka. Is there any maximum limit of the quantity of those two scavengers which could be acceptable from a regulatory perspective that can be added in the formulation without leading to a new bioequivalence study?
That's a great question. So I think in terms of maximum levels, I've not seen a maximum level being mentioned anywhere, and I've not seen either a level that would say, oh, if you have just that amount, you can let go of bioequivalence studies. So I think that's a conversation to be had with your regulatory body, and to have a conversation and a rationale as to why you would not need the bioequivalence study, for example. But of course, the level of inclusion, yes, is very small. And as the drugs get approved with certain levels of nitrate scavengers in it, there might be a point at which the regulatory bodies say, well, if it's really below 1%, below 0.5%, at this point, the bioequivalence is not needed. I mean, there has been studies like the one I mentioned by Dr. Bode looking at bioequivalence. He had 4 different drug models, class 3 drugs. And in his case, in his experimentation, which he was doing actually for the FDA, he was not seeing an impact. So if you can make the argument actually rationally that there is no reason that there should be an impact, and you can defend that, it might be that you can not have to do it. But again, it's a conversation with your regulatory body, with the data you have, to decide what's the best path to make sure you have a very safe drug product.
And if I may add, actually, you also can back up your reasoning with your in vitro testings, like when you do the in vitro dissolution testing and prove, and for certainly you will not have an impact on this testing of adding ascorbic acid. So it's another, let's say, element that you have in discussing with regulatory authorities that the bioequivalence would not be jeopardized.
There's definitely one question here that we can follow up later. Somebody asked, what is the LOD of our analytical method? So we'll get back to you with details on that. And then there is also a question around the particle size of ultrafine ascorbic acid. Do we have that information that we can quickly share?
Yeah, so this is a quite unique property of this product. It goes up to the 20 to, I'm not even sure, maybe I say a wrong number, 20 to 50 micrometers. So we'll go into the specifications, also share it, but this is, as mentioned, quite unique compared to the other products on the market, which have a quite significantly bigger particle size.
We can share definitely more details as well as a follow-up. And then somebody asked about whether there is a best ratio of ascorbic acid. Should it be linked to the nitrite content of the excipients, or whether there is any relationship between the concentration of ascorbic acid and the nitrites in the formulation in general, I suppose.
So yes, I can also take that up. First, in your formulation design, it is always recommended to use, of course, the excipients with low nitrate levels. And today there are also quite several on the market, which you can pick. However, then adding ascorbic acid is recommended because nevertheless, there will be nitrates present in the formulation depending on your processing parameters, especially if you have wet granulation. It would be good to have some safety net to make sure that there is less, actually that the nitrosamine formation is reduced or blocked completely. Now, when we come to the, let's say, prediction how much of the ascorbic acid should be added to the drug products, it will strongly depend on the drug molecule, but as well on the other excipients and process. So if you check numerous scientific literature recently published and always publishing on those effects and how the antioxidants work, they keep repeating you have to do the trials. And this is also what we have encountered when we started, and some minimum formulation design of experiments of few levels will be needed to make sure that you are there. What we definitely can say that those numbers are very low in percentages, so there is no point of testing 10-20% of ascorbic acid. It is quite low, up to 1. In our case, was even 1% enough. Can go even to 2, but in at least three trials are needed to optimize the best working concentration for you, and of course also to determine what is the lowest working one, because you do want to have the lowest level that is still efficient but not over added if not needed.
Is it better to use the salt or the free acid form of ascorbic acid? Any experience on that?
Yeah, so maybe also I can cover that. There is also possible to use sodium ascorbate as antioxidant in your formulations. It is recommended actually for the APIs that are pH sensitive. Of course, we know that there are some drug molecules that are from their nature sensitive for the acidic conditions. And if you want to prevent degradation of your API, you might opt for the sodium ascorbate instead of ascorbic acid, for example. But we do know that ascorbic acid has higher redox activity, so redox potential. So it is usually recommended if the drug substance will survive. In a way, adding ascorbic acid, it should be the first option.
Here's a quick one. Did you use added nitrite to your model studies or just endogenous nitrite in the excipients?
So depending on the study, for the study where we had placebo tablets, we did not add any nitrates. We had, and thank you for the question, it's quite important to think about, we have had a low nitrate excipients with low nitrate content, and we have checked those nitrate levels very much up under the 1 ppm levels excipients, and we still have in our tablets showed the increase of the nitrate levels, and when ascorbic acid was added, it was reduced. So it's in that case we didn't spike with nitrate, but for example other studies we have spiked to really show the effect of the nitrosamine formation, for example.
Is there any experience on different behavior of ascorbic acid with different vendors and manufacturers?
So for now we have taken, as you have seen, other products on the market, and we have tested their physical characteristics, but we didn't go into the studies using other also products.
Yeah, and what I can add on this one is we have seen differences as we've shown in the slides. There's some differences on impurities as well. We have heard from different customers about differences in behaviors and performance. Is very formulation dependent, of course, but there is indication that, of course, the particle size may play a role, but also the presence of impurities and how those ascorbic acids are manufactured can also have an implication. So it's an interesting question. There is always variability between different suppliers, even though they might look the same on paper. We all know that there might be some differences that are not on the paper, and we've seen some differences in the impurity levels of some of the ascorbic acid that we have analyzed from the market. So there is a potential difference for a behavior change or behavior difference there between the different suppliers. So just something to keep in mind, that it is possible to exist for sure. And Cecile, anything else you want to add on this? I jumped in.
Yeah, no, I think you covered it. But of course, for our own experimentation internally, we had access very easily to the dsm-firmenich material. So we didn't specifically studied other products on the market in tablets or in liquids. But as you said, yes, we've observed differences ourselves in the materials coming from different vendors. But now, yes, how it would behave. I think what's important for you is to have a very reliable source that is very controlled, so that you don't end up with material which changes over time.
And I think we'll do one last question. We have a lot of questions here on regulatory. We will take those and we get back to all of you with our best answer. But one that I know this is a sticky topic is the analytical method used to measure the nitrites and some of those nitrosamines. So somebody asked what type of analytical method was used for the nitrite testing reported. Can you just give a brief comment on what type of method was used? And we can share more details afterwards.
So yes, we have used HPLC analysis for the nitrate levels. Am I right, Cecile? Maybe I'm.
I think, yeah, it was UPLC and ultra perform. Yeah, UPLC mass spec. I think it's a method from Jaris, I think 2022. But we can double check because
Let's go to that.
Sretka and I are not the analyst performing the analysis of nitrite in particular.
We'll definitely get back to you on the details, but analytical method is a sticky topic on this nitrosamines. Okay, so I think we're out of time, but I do have all the questions here. So Dr. Parab, do you want to close out the session for us? And on behalf of the dsm-firmenich team, thank you so much for the opportunity. We've really enjoyed being here today. Hope this was useful. We'll get back to all the registrants with the follow-up questions when we have a chance. Thank you.
Thank you, Dr. Fernanda, and all the speakers, Dr. Ann, Dr. Mystic. So I think the IPEX team from the Secretariat will take over from now.
Yes. So on behalf of IPEC India, we extend heartfelt thanks to all the distinguished speakers, Dr. Anne Cecily, Dr. Zwalka, Dr. Fernanda, for your valuable insight and expertise. Dr. Fernanda for taking up the Q&A. All your contributions have enriched the knowledge of the participants. Sunali from Netherlands for facilitating this webinar. Akanksha from DSM Mumbai for all the coordination. Dr. Rajesh Parab, thank you very much for delivering the opening remark. ACG for allowing us to use the Zoom platform. Mr. Kaushik Desai for your guidance. Without it, it wouldn't have been a great success. Deepak and Dipesh for all the back-end work towards the webinar. Last but not the least, we are immensely grateful for all the attendees for their active engagement and questions. This might be the webinar where we had the maximum number of questions, and whichever are not answered, we'll ensure that we will revert to you all shortly. IPEC India will be having this annual conference in Hyderabad on May 31st, 2024. Request everyone to register, and you can get more information on our website and the LinkedIn page. Also, you may contact the Secretariat if you need to have any more details. Thank you, everyone, making this webinar a great success.
Thank you, Prema.
Thank you very much.
Thank you very much, Team DSM.
Bye, everyone.
Bye.
Thank you, everyone. Bye.